Please sit down. On behalf of the Vice Chancellor, Professor Adebayo Simeon Bamire, I welcome all of us to this 369th inaugural lecture to be delivered by Professor Olusanjo Adewale. Professor Olusanjo Adewale graduated from the University of Ife, Ile-Ife, with second-class honors upper division degree in biochemistry in 1987. He had the highest cumulative grade point average in his class. After his National Youth Service Corps, he enrolled and completed his Master of Science and Doctor of Philosophy degrees in biochemistry from Obafemi Awolowo University, Ile-Ife, in 1992 and 2003, respectively. He joined the services of Obafemi Awolowo University in 1999 as a graduate assistant and rose through the rank to the post of professor in 2014. Professor Adewale has received several distinctions and awards, some of which include European Molecular Biology Organization Short-Term Fellowship in Germany 2007, Carlstorm International Diversity Fellowship, United States of America in 2011, 2004 Dako Afolabi Most Productive Science Scholar of the Year Award. In 2011, he received European Molecular Biology Organization Bursary Award for HEMBO Practical Course in Computational Biology at the European Bioinformatics Institute in Easton, Cambridge, UK. He was a guest scientist at the Institute of Molecular Biology and Biotechnology of Plants in Germany. Over the years, Professor Adewale has focused his research in the following areas: enzymology, protein chemistry, molecular biology, and biotechnology, and has supervised 35 Master of Science and 5 PhD degree theses by research. He has to his credit 40 articles published in peer-reviewed journals. He also has 1 patent. He has participated in several conferences in Africa, Europe, and United States of America. He has served on several committees in the university and was head Department of Biochemistry and Molecular Biology between 2017 and 2021. He is the current Dean of the Faculty of Science. He was Editor-in-Chief, Biological Sciences. He was Editor-in-Chief, Biological Sciences of the Ife Journal of Science between 2017 and 2021. He has been serving as a reviewer for a number of journals published by Elsevier, Springer, Taylor and Francis, among others. He has been a resource person for National Board for Technical Education, NBTE, for resource inspection and accreditation of ND/HND Science Laboratory Technology, Chemistry and Biochemistry programs for several polytechnic and polytechnics in Nigeria. He served as collation officer for Ilaje local government in 2020 in Ondo State governorship election. He is a member of many professional societies, including Nigerian Society of Biochemistry and Molecular Biology and European Society for Comparative Physiology and Biochemistry. So I present to you Professor Isaac Adewale, who is going to deliver the 369 inaugural lecture titled An Odyssey into the World of Enzymes. Thank you very much.
Vice Chancellor, sir. principal officers of the university, royal fathers, we have in our midst His Royal Majesty Oba Engineer Timothy Adetoro, distinguished personalities, members of the university community, visitors, ladies and gentlemen. It is my pleasure to welcome you to today's inaugural lecture. It is the 5th of such in the Department of Biochemistry and Molecular Biology, Faculty of Science, and the 369th in the history of the university. The title of the lecture is An Odyssey into the World of Enzymes. An Odyssey is a journey made up of several twists and turns, full of excitements and also of disappointments. I will, however, reserve this latter part for another time. Enzymes are biological macromolecules capable of accelerating the rate of reactions by factors as much as 1 million or more over the uncatalyzed reactions and are themselves not consumed in the process. Enzymes drive processes of life in all living things because most reactions in living things do not take place at an appreciable rate without enzymes. With some exceptions, living things are organized structures capable of life forms, movement, reproduction, nutrition, response to stimuli, growth, and excretion. And all these functions are made possible through the catalytic activities of enzymes. Life is about chemical reactions, and chemical reactions in living organisms are about life, all of which are catalyzed by enzymes. To simplify the points even further, as a result of different enzymes working in tandem, I'm able to stand before you and speak. And you're able to sit down and listen because the food we ate had been digested and converted to monomeric units, which had been broken down to generate high-energy electron carriers, reduced fluffing adenine dinucleotide, FADH2, and reduced nicotinamide adenine dinucleotide, NADH. These are in turn being oxidized to produce adenosine triphosphate, the universal currency of energy in living cells. Hydrolysis of ATP by an enzyme called adenosine triphosphatase generates energy powering all the processes. In terms of application, the detergent with which you wash your clothes contains enzymes to remove stains. Malt drinks, wine, beer, yogurt, and bread are some of the products of enzymes in action. My journey into biochemistry as a discipline is probably by divine providence, as I never had a permanent ambition for any course of study. My ambition oscillated between science and art-based disciplines. I must, however, admit that I enjoyed chemistry because it appeared magical or more adventurous. A glowing splinter could burst into flames magically when deep inside a tube. The flame of a burning splinter could be extinguished by dipping it into another tube containing an invisible gas. Experiments performed by our general science teacher in year 1 secondary school. I eventually developed interest in biochemistry when my brother, Dr. Adewale, who had developed a keen interest in my educational development, enrolled me in the then Oyo State College of Arts and Science, Oscars Ilefe, for advanced level program in biology, chemistry, and mathematics. It was at this stage that it struck me that activities in living cells could be explained using chemistry. That the gap between chemistry, the study of structures and interactions of atoms and molecules, and biology, the study of structures and interactions of cells and organisms could be fused. I got fascinated by the realization that since living things are composed of inanimate molecules, life at its most basic level is a biochemical phenomenon. I came across terms like Krebs cycle, urea cycle, glycolysis, purines and pyrimidines, cholesterol, essential and non-essential fatty acids, etc. at this stage. After my first degree, the Obafemi Awolowo University and Professor Adenika Folanyan, who was then the head of the Department of Biochemistry, in the now Department of Biochemistry and Molecular Biology, Faculty of Science, midwifed my postgraduate training, leading to a PhD degree in biochemistry in 2003. Obafemi Awolowo University provided a monthly stipend of 250 naira for the Master of Science degree program, graduate assistantship and assistant lectureship for the PhD training, while Professor Adeyinka Folanyan provided the needed mentorship. The chemistry of life. The living matter contains elements that were selected according to their abilities to perform certain structural functions or to provide specific reactivities. Cells contain 4 families of small molecules or their derivatives, amino acids, nucleotides, sugars, and fatty acids. Each of the families performs different functions and can be used as building blocks of larger molecules which are called polymers, amino acids. There are more than 100 amino acids found in nature, but only about 20 amino acids are needed to synthesize proteins in most living organisms. These 20 amino acids are all L-isomer alpha amino acids. All of them, except glycine, contain a central chiral carbon. The amino acids selenocysteine and pyrrolysine are considered the 21st and 22nd amino acids, respectively, and are more recently discovered amino acids which may become incorporated into protein chains during ribosomal protein synthesis. The general structural formula for amino acids is as projected on the wall. Each amino acid contains an amino group protonated under physiological condition, a carbonyl group which is also ionized, and a side chain designated R group. Amino acids are used primarily in the synthesis of long complex polymers known as polypeptides and are joined together by peptide bonds. Short polypeptides up to a length of about 50 amino acids are called peptides. Longer polypeptides are called proteins. Proteins are involved in catalysis as enzymes, the theme of this lecture. Enzymes. The word enzyme was coined by German physiologist Wilhelm Kuhn in 1878 from the word hen, yeast, zyme. Hen, yeast, zyme. So enzyme, hen, yeast, to describe alcoholic fermentation of sugars by yeast cells. In 1897, 2 German brothers, Hans and Edward Buchner, ground a slurry of yeast cells with sand, obtained a cell-free extract by filtration, and then attempted to preserve the extract with sugar syrup, only to notice fermentation. He named the extract Zymes, thus demonstrating that yeast extract could catalyze reactions in the absence of yeast cells. It was later realized that they are present in all life forms and have a remarkable properties and are capable of increasing reaction rates by factors of between 1 million and 1 trillion. They are able to perform this feat under mild conditions of pH and temperature in which cells function due to their complex structures. To illustrate the power of enzymes, consider the chemical synthesis of ammonia by Haber process. That's Haber-Bosch. A mole, as shown on the board, a mole of nitrogen combines with 3 moles of hydrogen to form 2 moles of ammonia. This reaction can only take place at the temperature of 450 to 500 degrees Celsius, a pressure of 200 atmosphere in the presence of reduced iron as a catalyst. This same reaction can be catalyzed by an enzyme called nitrogenase, which is present in nitrogen-fixing bacteria. A particular genus, Azotobacter, is present in the root nodules of leguminous plants and helps convert atmospheric nitrogen to ammonia, which in turn is taken up and used and utilized by the plants. The reaction takes place at ambient temperature and pressure, that's 25 degrees Celsius and 1 atmosphere, respectively. An extreme example is orotidine 5'-phosphate decarboxylase. This enzyme catalyzes a reaction which will take millions of years to occur in milliseconds. It must be stated that at this point, that not all enzymes are proteins. It is now known that some RNA molecules termed ribozymes are catalytically active and play central roles in the 3 kingdoms of life. They have been found to play roles in tRNA maturation, intron splicing, replication of RNA viruses, and protein synthesis. However, the vast majority of enzymes are proteins. Properties of enzymes. Enzymes are highly specific, both in the reactions they catalyze and in the choice of reactants, which are called substrates. They are also sensitive to temperature and concentration of hydrogen ion, that's pH, of solution, where the enzyme is contained. Ionic strength of the solution, metals, organic or inorganic ions present in the solution, which may serve as inhibitors or activators in some cases. Class 4, lyases, class 5, isomerases, class 6, ligases, and class 7, translocases. Applications of homogeneous enzyme preparations isolated from various organisms. Enzymes are used to make or improve nearly 400 everyday consumer and commercial products, as shown in tables 1 and 2. The processes use less energy, water, and raw materials, and less waste is generated. The application can be broadly divided into medical or clinical use as drugs, food and beverage processing, animal nutrition, textiles, household cleaning, bioremediation of crude oil contaminated soil, and in mineral exploration. Medical applications. Table 2 is a summary of the main pathologies and conditions that can be treated with enzymes isolated from various organisms, including bacteria, fungi, plants, and animals. The table shows that in clinical applications, enzymes can be used to treat lysosomal storage diseases and many others. Herbicides, environmental contaminants, and conjugation with glutathione makes the cyanobiotics more water-soluble and can then be eliminated from the body via the mercapturic acid pathway. Adewale and Afolanya, 2004. As a result of the activities, they have implicated the resistance to drugs in humans and resistance to herbicides and pesticides in plants and insects, respectively. They are often expressed in organisms living in polluted environment. Organisms could cope in polluted rivers. Aquatic allelochemicals, as shown in figure 5, was attributed to the unique biochemical properties of GST that they have. Adewale and Afolanya, 2006, Timmy and Adewale, 2021. Furthermore, we have speculated that the hardiness of extrapolation of one parameter, X, time of storage, if the other one, Y, specific activity is known. The results obtained also permit us to suggest that measurement of residual activity of GST in frozen meat or fish samples could provide information about storage history and, by extension, the freshness of the items. Our work on glutathione transferases was sponsored by grants received from the University Research Council, URC, of the Senate of Obafemi Awolowo University, as well as institution-based research grants of Tertiary Education Trust Fund. Aldehyde dehydrogenase. Aldehyde dehydrogenases are a group of enzymes that catalyze the conversion of aldehydes to their corresponding carboxylic acids, and they usually use either NAD, that's nicotinamide dinucleotide, or nicotinamide dinuclear phosphate as a coenzyme. They are also ubiquitous, being present in plants and animals. Our interest in plant aldehyde dehydrogenase, LDH, arose because of their role in stress tolerance in plants. Plants have evolved a variety of definite strategies to remove reactive oxygen species, ROS. ROS accumulation to an excessive amount could cause disruption to cellular machinery and homeostasis. The harmful effect of excessive amount of aldehyde is well established, and they are directly involved in various illicit reactions, such as compromising the integrity of membrane lipids, nucleic acids, or proteins to form adducts, which alter their functionality and manifest phenotypically in form of cell tissue death, organ failure, tumor formation, etc. Family 3 aldehyde dehydrogenase is an important determinant of resistance to toxicity mediated by intermediate chain length aldehydes that are produced during lipid peroxidation. Overexpression of class 3 aldehyde dehydrogenase isoenzymes in Arabidopsis thaliana cause more than 30% increase in tolerance to drought and high sodium chloride concentration in the laboratory of one of my German collaborators, Professor Dr. Dorothea Bartels of the Institute of Molecular Biology and Biotechnology of Plants, INBAO, minus 20 degrees Celsius, instead of less than a day before my joining the group. One notable outcome of the study, in addition to stability, was that the chain length of the substrates has a significant effect on the activity of the enzymes. Heptana, nonana, and decana were the substrates with the highest specific activity. The enzymes were also inhibited by product of the reactions they catalyzed. The work was published in Biochemical Journal, London, one of the most prestigious biochemical literature in the world. Stettel et al., 2011. Amylases. Amylases are a group of enzymes that catalyze the hydrolysis of starch, producing oligosaccharides, notably maltose, glucose, maltotriose. They hydrolyze alpha-1,4 and alpha-1,6 glycosidic bonds in starch and are classified into different groups, namely alpha-amylase, alpha-amylase, also called 1,4-alpha-D-glucanohydrolase, are widespread in nature, being found to be present in most organisms from bacteria, fungi to man. They are metalloenzymes as most require calcium for activity and they hydrolyze starch from non-reducing end, producing smaller fragments and can thus reduce the viscosity of starch slurry, that is liquefied gelatinized starch. They are thus valuable industrial enzymes and are extensively used in food industries, breweries, bakeries, and confectionaries. They are also used in laundries. Beta amylase, also called 1,4-alpha-D-glucan-maltohydrolase, is present in bacteria and fungi and plants. It hydrolyzes starch or oligosaccharides produced by the action of alpha-amylase to produce maltose, starting from the non-reducing end. The presence of alpha-amylase and beta-amylase in a suspension of the right pH and temperature may lead into the hydrolysis of such starch, producing sweetened sugary solution together with limited dextrose. It is therefore used extensively in many applications like alpha-amylase. Gamma amylase, also called glucosidase, amyloglucosidase, exo-1,4-alpha-glucosidase, can hydrolyze alpha-1,4 glycosidic bonds at the non-reducing end of starch, comprising amylose and amylopectin, as well as alpha-1,6 glycosidic bonds present in amylopectin yielding glucose. Industrial uses of amylases in Nigeria are widespread in the breweries to break down cereal, that's maize sorghum, starch into fermentable sugars. In beverage manufacture, that's production of high glucose maltose syrup. In laundry to remove starch, and also as additives in detergents to remove stains. Increasingly, they are also used in biofuel production to degrade starch fungal amylases with amylases obtained from local grains, particularly sorghum and millet. Adewale et al. 2006, Adewale Oladejo 2009, Adewale et al. 2012. This is because bacterial fungal amylases used in Nigeria are expensive and are imported using scarce foreign exchange. We have malted the grains essentially using locally fabricated materials, killed the grains, extracted and purified amylases from the grains using conventional purification procedures, and shown that the sorghum amylases can be used as substitutes. That killed malted sorghum can be stored for several months with the amylases still intact for further application. Mr. Vice Chancellor, sir, I introduced the word unkilled into the Dictionary of Science as an alternative to grain malt that have been used for over a century. In the process of amylolytic enzyme production and characterization, we have optimized the condition for maximal amylase production. When sorghum grains are steeped in water adjusted to pH 6.5 at 20 degrees Celsius, 8-fold higher amount of amylolytic enzymes are produced. Adewale et al. 2012. Furthermore, we have used amylolytic enzymes from sorghum grains to break down cassava starch into fermentable sugars, as will be further enumerated in the course of this lecture. Invertases, also known as beta-fructosidases or beta-fructofuranoidases, are found in yeast, bacteria, fungi, and other organisms, such as bees, and also present in higher plants. Invertase catalyzes than the sucrose, and so less of it is required to sweeten our drinks. It is therefore used in large quantity in food industries, confectionaries, ethanol production, drugs, and cosmetics. Invertase is mainly sourced from yeast. My interest, again, stems from the belief that invertase from locally sourced yeast from our environment could replace the imported invertase, thus saving the country foreign exchange. We have screened palm wine from several sources in southwestern Nigeria for yeast that can produce invertase in large quantities. To my surprise, yeast, Saccharomyces cerevisiae, from Abagboro village. Abagboro village is about 500 to 1 kilometer from here. produce the highest amount of phyphatase and the highest concentration of ethanol compared to Burras yeast obtained from a multinational brewery in Nigeria and other local yeast strains within the southwestern Nigeria, as well as other commercially available yeasts. Phyphatase from Abagboro yeast, Saccharomyces cerevisiae, has been characterized in my laboratory. Mr. Vice Chancellor, sir. To fully demonstrate the applicability and domesticate the technology, we got cassava tuber from local farmers as a source of starch, mashed it into slurries, gelatinized the starch, and used sorghum grain amylotic enzymes to liquefy and saccharify the starch to obtain a sugary fermentable solution. Using locally sourced soybean as a source of nitrogen for the sugary solution, we fermented the solution called wort using Abagboro yeast to obtain 24.2% ethanol compared to 16.7% for yeast from hydroxyphenyl ethylamine, otherwise known as dopamine. These reactions have been the subject of intense investigations in the last 3 decades or so because of their applications in several biotechnical processes. Some of the processes include production of 3,4-dihydroxyphenyl dihydroxy L-phenylalanine, L-DOPA, a drug widely used to treat Parkinson's disease, and production of cross-linked protein networks for use as novel food additives. It is also an oxidase that is directly involved in the production of melanin, which is responsible for the color of our skin. Inhibitors of tyrosinase are used in various cosmetics as skin whitening agents. In plants, tyrosinase is responsible for branding reactions observed in apple, yam, etc. because of the oxidation of phenolics found in such items. The enzyme has traditionally been sourced from mushroom Agaricus bisporus. My interest in tyrosinase research stems from the fact that different species of yam could serve as a cost-effective source of tyrosinase for various industrial uses. We devised a novel method of purification of tyrosinase from 4 species of yam, namely Dioscorea praensilis, Dioscorea alata, Dioscorea rotundata, Colocasia esculenta, otherwise known as cocoyam, which were found to contain highest tyrosinase protein than other tubers such as Xanthosoma mafafa, Apomia batatas, and Dioscoreum lumentorum. Tyrosinase from the 4 species was purified to homogeneity as projected on the board in figure 7. The homogeneity was measured by sodium disulfate polyacrylamide gel electrophoresis. We showed that all the purified enzymes were activated in 40% eta by between 120 and 170%. Furthermore, they maintained 100% residual activity in 7% eta for 17 hours. Optimum pH and temperature were 6.5 and 50 degrees Celsius, respectively. Beta-macrobutadienol and glutathione were inhibitors of the enzyme. We showed that different color products, figure 8, which shown in figure 8, could be formed in aqueous environment in the presence of substrates or additives, including catechol, pyruvic acid, and ammonia. Olusanjo et al. 2014, Olusanjo et al. 2021. Mr. Vice Chancellor, sir, this piece of information is valuable for organic and pharmaceutical chemists for the green synthesis of drugs and novel food and pharmaceutical compounds. We have extended this study to Moringa oleifera leaves as a source of this enzyme, Agumbiade and Adewale 2022. Interestingly, there was a spike in the international demand for Nigerian yams when we first published this work in 2014. In a study quite similar to tyrosinase work, we also showed that white and red cultivars of kola nut, Kola nitida, could be a source of peroxidases, which are also used in various diagnostic reagents as reporter enzymes. Adewale and Adekunle 2018. The fund for this work was provided by TETFON institution-based research grants. Serine proteases. Serine proteases are found in many living cells as part of molecular scissors, responsible for cutting unwanted proteins in food digestion to break down the polymeric proteins into peptides and amino acids to facilitate absorption, or in post-translational modification of proteins to generate new functionality for roles in cell proliferation, immunity, and inflammation. Aberrant activity, otherwise known as hyperactivity or inactivity, or some of the proteases have therefore been implicated in many diseases, including cancer, cardiovascular disorders, autoimmune diseases, and viral infections. We have focused our attention on two proteases known as furin and TMPRSS2, which is transmembrane serine protease 2, which have been implicated in several human pathologies, including cancer, HIV/AIDS, and COVID-19. Furin is a member of evolutionary ancient family of protein convertase and has been termed master switch of tumor growth and progression, as its aberrant expression or activation can promote the formation and progression of various malignancies, as shown in figure 9 on the board, and are involved in the infectivity of SARS-CoV-2, that's severe acute coronavirus 2. Several studies have shown that SARS-CoV-2 spike proteins are cleaved by host cell furin at the S1, S2 site, thus allowing the virus to enter its genome into the host cell. Brand and Salter, 2019. TMPRSS2, on the other hand, has been an important target in the treatment of seasonal influenza infections and contributes to prostate carcinogenesis and metastasis. In order for SARS-CoV-2 to enter cells, TMPRSS2 has also been implicated has been responsible for cleaving the spike proteins at the S2 site. The studies suggest that inhibitors of these proteases have high therapeutic potential for treatment of these diseases. Based on a report by Mujunda et al. 2010, many medicinal plants had hitherto been thought to exhibit their medicinal properties by virtue of high polyphenols and flavonoids present in them, actually contain inhibitors of serine proteases such as protein convertases. With international funding support from 7 funding agencies, namely Science Granting Council's initiative in Sub-Saharan Africa and administered by South Africa's National Research Foundation, NRF, in collaboration with Canada's International Development Research Center, IRC, the Swedish International Development Cooperation Agency, SIDA, South Africa's Department of Science and Innovation, DSI, the funds the researcher, the Quebec FRQ, the United Kingdom's Department of International Development, DFID, United Kingdom Research and Innovation, UKRI, through the Native Fund, and the SGCI participating councils under the COVID-19 African Rapid Grant Fund, HI, as the principal investigator, in collaboration with Professor Dr. Daniel Sauter of the University Hospital in Tübingen, Germany, and other partners from University of Johannesburg, South Africa, and Obafemi Awolowo University, screened for medicinal plants. Momodica charantia, in English called bitter gourd, in Yoruba is called ejirin wewe. Lawsonia inamis, in English called enna, in Yoruba called lali. Xylopia ethiopica, in English called guinea pepper, in Yoruba is called eru. And Iminocardia acida is called urupa. For inhibitors that are single molecules capable of inhibiting the proteases. Extracts from these plants have traditionally been used to manage at least 4 viral diseases. Adewale et al. 2023. Results from our laboratory suggest that the plants have phytochemicals. Some of them are serine activators of serine proteases, as shown in figure 10. We are currently carrying out studies to determine whether the purified extracts could block the replication of HIV and SARS-CoV-2. If the inhibitors are found to inhibit SARS-CoV-2, they could be further developed into drugs to manage COVID-19. Mr. Vice Chancellor, sir, I'm also happy to announce to you that my collaboration with the NanoBio Laboratory of Professor S. Oluwafemi, University of Johannesburg, South Africa, and Authority Nanoscience and Technology is yielding positive results, as we have observed for the first time that quantum dots could serve as inhibitors of serine proteases, as shown in figure 11. Administrative and community service. Mr. Vice Chancellor, sir. In addition to research engagements and teaching, I've also been involved in various activities within and outside this community. At the departmental level, I have successfully supervised and mentored 29 MSc and 5 PhD students by research, and served in almost all departmental committees at one time or the other. I was acting head of department between 2017 and 2019, and head of department between 2019 and 2021. I was chairman of the first Faculty of Science Conference 2004, and also chaired it between 2016 and 2021. I was chairman technical subcommittee of the conference between 2005 and 2015, serving under Professor M.A. Eleruja. I was editor-in-chief of FIFA Journal of Science between 2017 and 2021, together with Professor A. Ogunfowokan, during which time the journal became truly international. As representative of Faculty of Science, I was a member of the Faculty of Pharmacy Review Panel between 2014 and 2016, Faculty Board of Social Sciences between 2017 and 2019, Faculty of Basic Medical Sciences Review Panel between 2019 and 2021. I was elected unopposed as Dean, Faculty of Science in 2021, and still serving as Dean of the Faculty. I've served as University Examiner for the award of MSc PhD degree, apart from my department, in the Department of Pharmaceutical Chemistry, Faculty of Pharmacy, Animal Sciences, Crop Production and Protection, Faculty of Agriculture, Immunology and Hematology, Faculty of Clinical Sciences, Chemical Engineering, Food Science and Technology, Faculty of Technology, Departments of Botany, Zoology, Chemistry and Microbiology, Faculty of Science. I was elected Senate representative in the Library Committee between 2019 and 2020 and served as internal assessor for promotion to the post of reader professor in the Faculty of Agriculture. Outside the OU community, I've served as external assessor for promotion to the post of reader at the University of Medical Sciences, UNIMED Ondo, external examiner for the award of MSc degree in the Department of Biochemistry, Federal University of Technology, Akure, and Osun State University, Osogbo, external examiner for BSc Biochemistry degree program of the Federal University of Yekiti between 2018 and 2021, external examiner at the Department of Chemistry and Integrated Science of the University of Ibadan, degree program at Osun State University, Osun State College of Education, Ilesha, between 2020 to date. Again, at the national level, I have contributed to the development of science and technological education in Nigeria by serving as resource person for National Board for Technical Education, for resource inspection, accreditation of ND, HND, Science, Laboratory Technology, Chemistry and Biochemistry programs at Niger State Polytechnic Zungeru, Niger State, that was in 2009, technical colleges in Osogbo and Ile-Ife between 2009 and 2010, Federal College of Freshwater Fisheries Technology, Nibusa, Niger State, Nigerian Army School of Military Engineering, Makurdi, Benue State, Abubakar Tatari Ali Polytechnic, Bauchi State, Ibarakwa Polytechnic, Iruwa, Oyo State, Delta State Polytechnic, Otefe, Ogara, Delta State, Heritage Polytechnic, Court, Udota, Eket, Akwa State. I served as Ilaje Local Government Coalition Officer for 2020 Ondo State governorship election. At the international scene, I'm a reviewer for analytical letters published by Taylor and Francis, Fish Biochemistry and Physiology published by Springer Verlag, Preparative Biochemistry and Physiology, Bioactive Carbohydrates and Dietary Fiber, Ecotoxicology and Environmental Safety, Comparative Biochemistry and Physiology, all published by SFR and PLOS ONE and several others. I facilitated as Dean of Science collaboration agreement between Obafemi Awolowo University and the University of Johannesburg, South Africa. The memorandum of understanding was signed in South Africa on 31st of January 2022. We have initiated an arrangement to extend the collaboration to Vaal University of Technology, South Africa. I am currently engaged in international drug discovery research collaboration between my research group and the laboratory of Professor Dr. Daniel Sauter and Professor S.O. Oluwafemi, respectively, of the Institute of Molecular Virology, University of Tübingen, Germany, and Nanobio Laboratories, Biochemical Sciences, University of Johannesburg, Africa. Apart from research, the international collaboration has been fruitful, as the faculty has received equipment and reagent donations, as well as fund for the institution of faculty prize for students with the highest cumulative grade point average from the University of Johannesburg through the professor. One of our PhD students just returned from the University of Tübingen on a student exchange visit. In 2022, as Dean, I initiated and facilitated the collaboration between Obafemi Awolowo University, Pan-African University of Life and Art Sciences Institute, Policy, University of Ibadan campus on one hand, and the Montfort University, Leicester, United Kingdom, on a British Council-sponsored circular plastic economy innovation hub that stores students of faculties of science and technology competing with others from University of Nigeria, Nsukka, University of Lagos, Ahmadu Bello University, Zaria, on innovations to eliminate plastic wastes in the environment. In this project, coordinated by Dr. Sahid Adekunle of the Department of Chemistry, Ms. Akeremi Olubomi Josephine of our Computer Science and Engineering Department eventually emerged winner and represented Nigeria at the Commonwealth Head of Government meeting held on 22nd and 23rd June 2022 in Rwanda. Recommendations. Vice Chancellor, sir. The study of enzymes as catalysts within living cells and in vitro outside is a cornerstone of development in medicine, agriculture, biotechnology, and even in green chemistry. In medicine, it was stated that earlier that enzymes may be derived from bacteria, fungi, plants, or animals, or produced by recombinant DNA technology, could be taken directly as drugs for some diseases, lacking the enzyme for a particular reaction, or to break down a particular substance in the body, e.g. streptokinase, which breaks down blood clots in some cases of heart attack, otherwise known as myocardial infarction and embolism, glucocerebrosidase to manage Gaucher's disease, or recombinant deoxyribonuclease to manage cystic fibrosis. Except for industrial enzymes, which don't necessarily have to be homogeneous, most of the enzymes in chemical and biotechnological applications are supposed to be used in pure form. Otherwise, unwanted side reactions may occur. Enzyme purification and characterization are an expensive venture that requires high level of skills. No wonder biochemists in the field of enzymology have moved to other less financially demanding areas. Thus, the school authority should consider this area of research for funding as it was in the beginning when IFE was the foremost institution with facilities for enzyme or protein chemistry research. Our molecular biology program in the Department of Biochemistry and Molecular Biology, probably the first in the country, has not taken off since council approval about 8 years ago due to lack of funds. I recommend that the university should consider funding our molecular biology program a priority. In the process, enzymology will benefit for they are closely integrated. To borrow a leaf from other countries who have recognized enzymology as key to biotechnology development, Nigeria as a country can establish Institute of Protein or enzyme research in different parts of the country. This is the practice in developed countries such as Germany, Japan, United States of America, Russia, to mention but a few. Alternatively, Obafemi Awolowo University can take the bull by the horn by kickstarting such. Between 1974 and late 80s, the Department of Biochemistry was so well equipped that our graduates who chose to pursue further studies in Europe or America had a seamless transition because they had been exposed to all necessary tools similar to what obtained in those climes. There was no university webometric ranking then, but we were getting recommendation letters from Ivy League schools in Europe and America. Biochemistry or molecular biology is the language of life sciences. Allocating resources to biochemical research will stimulate high-quality, cutting-edge research in other life sciences, including medicine and agriculture. Researches such as the creation of high-yielding transgenic crops, able to withstand drought or salt stress, diseases and pests, production of novel foods and nutraceuticals, novel drugs for old and emerging diseases, novel catalysts for biotechnological applications, etc., can be done routinely here in IFE if you are well equipped and will to publications in high-impact journals, improving the image of the university in addition to solving problems in our immediate environment. Acknowledgements. I will start this appreciation note by acknowledging my late parents, Madam Abigail Adewale Bewaji, who provided an unlimited degree of freedom. Apart from some of the don'ts of Exodus chapter 20 in the Holy Bible, I was free to do what I liked. My father, late Chief Joseph Adewale Bewaji, was a disciplinarian and permitted limited degree of freedom. These two extremes probably molded my character and allowed me the ability to be at peace with others with diverse opinions, culture, and religion. I appreciate all my brothers, particularly Dr. Ademola Adewale, Bishop Mike Adewale, and late Honorable Dr. Elijah Adewale, Dr. Adewale Adewale should be seated somewhere around that place. They took an unusual interest in me since I was about 10 years old and guided me even up to this moment. I gained knowledge and wisdom from them. I acknowledge the contributions of my former teacher and supervisor, Professor Adenika Folanya. He was responsible for the mentorship of my PhD program in biochemistry and gave me a cash donation of 3,000 naira for the payment of publication charge of my first and last article, a journal of Nigerian Society for Biochemistry and Molecular Biology. He recommended me for the award of Obafemi Awolowo University Postgraduate Fellowship Award, which enabled me to enroll and complete my MSc degree in biochemistry. I appreciate my former teachers in the Department of Biochemistry and Molecular Biology and Department of Microbiology, particularly Professor F.D. Onojobi, Professor A. Abodare, late Professor Baolalade Afolanya, and late Professor Shonukan for the showed love to me. I appreciate all my former lecturers in the faculty through whom I have gained knowledge. I appreciate all past days in faculty who had their assigned responsibilities to me, which enabled me to acquire requisite experience, which are now found to be very useful. Professor M.A. Badejo is seated here. So, Professor V. Olariwaju, Professor W. A. Muse, Professor A. Ogunfowokan, Professor O. Adedeji, and Professor M.A. Eleruja. I always enjoyed their company and the company of my good friends, Benga Libiowu and Akinroko Akifala, among others too numerous to mention. I appreciate all lecturers and non-teaching staff in the Department of Biochemistry and Molecular Biology, and in all the other departments and units in the Faculty of Science, Department of Microbiology, Botany, Zoology, Chemistry, Geology, Mathematics, Physics and Engineering Physics, and Center for Energy Research and Development. For the confidence they repose in me, God will continue to uphold us so I don't betray that trust. I appreciate Professor Simeon Bamere, who as Deputy Vice Chancellor Academics, encouraged me to apply for an international research grant funding during the COVID-19 era. I heeded to his advice. He called me around 4 a.m. one morning to break the news of the success of the application. It appeared he was even happier than myself on the success. I want to appreciate my international collaborators. Professor Samuel Oluwatobi Oluwafemi has been wonderful, hardworking, and God-sent. Through him, the university and the country stand to gain tremendously, for he is the leading expert in nanoscience in the world. I appreciate Professor Dr. Daniel Sauter of the University of Tübingen, Germany, and Professor Dr. Dorota Bates of the University of Bonn, Germany. I appreciate all my present and former students and all other collaborators in Obafemi Awolowo University, Professor Mrs. O. Adedeji, Professor A. Folonshaw, Professor M.A. Aderogba, Professor B. Omafube, Professor E.F. Aransiola, Dr. A. Fudeke, and late Dr. M.K. Bakari. I appreciate all the agencies that have been funding my research work: University Research Council of the Senate of Obafemi Awolowo University, Ile-Ife, European Molecular Biology Laboratory, European Molecular Biology Organization, EMBO, Heidelberg, Germany, TETFund, Institute-Based Research, and all the 7 organizations that funded the COVID-19 African Rapid Grant Fund. Last but not the least, I appreciate my wife, Mrs. Margaret Omolade Adewale, seated somewhere around here. And the children, Grace, Ibukun, Hayo, and Adeleye. My wife has been the one keeping the home front while I engage in teaching, research, and community service. I appreciate the love of my mother-in-law. My mother-in-law is also seated in this gathering. Chief Mrs. Victoria Boladale Adenipekun. She's one of the most intelligent women of our generation and a Christian activist. I appreciate my brothers-in-law and their families. I especially appreciate all my mentees friends, my inaugural lecture planning committee for their efforts and useful suggestions. Thank you for your kind attention. Thank you, ma'am. Thank you. Thank you very much.